Linkage and crossing over. Genes which are found on the same chromosome, if they are closely placed, they are said to be linked. Linked genes inherit together in the form of blocks. If we define the linkage, genetically, linkage can be said that it is the failure of the segregation of the chromosomes, or it is the failure of the segregation of the genes, because the genes, they are closely placed, so during the meiosis they will not segregate from each other. Linkage is failure of segregation of the genes during meiosis. Linkage is based on the frequency of the heterozygotes. If the genes, they are far from each other, there will be higher frequency of the heterozygotes or the recombinants. If the genes, they are close to each other, there will be less frequency of the recombination. So if there is less frequency of the recombination, we can say that genes, they are linked with each other. Crossing over is a phenomena that takes place during meiosis 1. During meiosis, homologous chromosome, they align with each other. During this alignment, the homologous chromosome, they exchange the fragments of the chromatids with each other. This exchanging of the fragments is between the non-sister chromatids of the homologous pair. This exchanging the parts of the fragments or the certain parts in the form of genes is called as chiasmata. Chiasmata formation is like a symbol of X or cross that represents that the fragments of the non-sister homologous chromosome, they exchange the fragments. In this diagram, we can see that how the fragments are changed during the process of meiosis. This is double-stranded DNA of the mother. This is double-stranded DNA of the father. During the process of meiosis 1, where chiasmata formation, a nuclease that digests one of the strands, by digesting the strands, these non-sister chromatids, they will exchange the parts. Here we can see that here the parts, they have been broken from this location, and here is the ligation, DNA ligase, that can join the fragments to the non-homologous chromosome. Here we can see the original color was red, but the fragment of the blue was attached to red color. So as a result, we can see the original were red, double-stranded DNA in red color of the mother, and double-stranded DNA of the father was in blue color. But here we can see that the chromosomes, they are in the form of recombinants. This is due to the chiasmata formation, or this is due to the crossing over between the homologous chromosomes. In this diagram, we can also see that how homologous chromosome, they can exchange their fragments. This is a pair of homologous chromosome. These are the chromatids, while during the meiosis or during prophase 1 of the meiosis, they break and cross over their fragments. So this is yellow color, this is blue color, but as a result of breakage and reunion, the fragments of this homology has been attached here, while the fragment of this homology has been attached here. This phenomena is called as crossing over, and this symbol is called as, that is like the X, is called as chiasmata formation. In this diagram, we can see that how crossing over in prophase 1. If there is independent assortment of two genes, they have independent assortment, then it means there is no crossover between the homologous chromosomes. In this diagram, we can say that one chromatid of this homologous chromosome is here, while the second is here. So because these are two different homologous pairs, between two different homologous pair of chromosomes, there will be no crossover, because the crossover is between a set of homologous chromosomes, between the one set of homologous chromosomes. linkage between two genes which are present on a single pair of homologous chromosome. Here we can see that this is a single pair of chromosome. It will segregate in this if the genes they are far from each other, then the gametes they will be the parental gametes. This is the parental gamete, parental gamete, parental gamete and parental gamete. So there is no exchange occur between because the genes they are located far from each other. But in this diagram we can see that there is exchange of fragments between non-sister chromatids. This is one chromosome and this is the second chromosome. Here we can see that each of the chromosome has sister chromatids. During a process of meiosis, these sister chromatids they have changed the fragments. Here is the chiasmata formation. Due to this chiasmata formation, the blue part has been attached with the yellow or brown part of the chromosome. So these are the parental. This is parental gamete, that is exactly like the DNA or the chromosome of the parent. This is also parental. While these two they are different from the parents because they have changed their fragments. As a result of chiasmata formation or as a result of crossover, here we can see that two of the gametes will have the parental type. This is parental type and this is parental type. P is for parental. While these two they are the non-parental type. So we can say that linkage and crossover are the phenomena they took place during the meiosis.